Welcome to Study Time, a televised home learning program produced by Rwanda Basic Education Board.
Hello students, a very warm welcome to the TV learning programs. I'm teacher Herbert. As usual, I will be taking you through the biology lesson. And the biology lesson of today is of senior 2 class. So the lesson of today is biology and health science. The class is senior 2. And we are going to be focusing on unit 5. And unit 5 is identification of food components. I think all of you know what are food components. The classes of food. Food components, the components of food. Components of food. We have the components of food. We have the carbohydrates. And I know some of you know what are the functions of carbohydrates. Carbohydrates are the energy-giving foods. They give us energy, and that energy is going to be used for doing work, for movement, all right? We have another component of food. We have, number 2, we have the proteins. Who can give me examples of proteins? Examples of carbohydrates, we have a number of food that contains carbohydrates and starch. Take an example like Irish potato. We have the cassava. These are some of the examples of carbohydrates. We have proteins, examples of proteins. We have fish. We have fish. We have beans. We have peas, and so on and so forth. We have another example, which is number 3. We can have lipids. Examples of lipids, we have fats and oils. Fats and oils. Fats are sold at room temperature, then oil are liquids at room temperature. So we have another class which is number 4. And we have the mineral salts. Mineral salts. We have a number of examples of the food which contains mineral salts. That's why we absence of those mineral salts in the food diet, someone suffers from a deficiency. Lack of calcium in the diet causes poor bone formation. Poor bone formation. We have examples. We have calcium ions. We have iodine. Iodine, that's another which is also needed in our diet. Absence of iodine in the diet causes goiter, swelling of the neck. We have number 5. Number 5, we have vitamins. A number of food contains vitamins, which is concerning about examples of vitamins. We have vitamins. We can talk about most of the fruits. Fruits are examples, vegetables and etc. etc. Let's now go to the previous lesson we discussed. Today we're going to be one which is lesson 2, which is concerning about testing of non-reducing sugars and proteins. I think, students, you do remember the previous lesson. The previous lesson was concerning about testing of the first lesson. Lesson 1 was concerning about testing of biological molecules, which was testing of starch and reducing sugars. Reducing sugars. If you do remember, we are going to go through this, how we did it. We talked about food they contain. How do you identify? And we say the food contains starch. Most of the starch is found in many food that we eat. It is found in cassava, Irish potato, sweet potato. All of them, they contain these food nutrients. Let's remind ourselves on lesson 1. We talked about the reagent that is used to test the presence of starch in a food sample. Who can remind me the reagent which is used to test the presence of starch? The reagent that can talk about reagent or chemical that is used to test the presence of starch in the food sample. Are you ready to tell me that reagent? Of course, the reagent that is used is iodine solution. Iodine solution. Iodine reagent or solution. Once you use the iodine solution, of course, you are testing the presence of starch in the food sample. When the food contains starch, what confirms the food contains starch? The color of iodine, which is brown in color, changes from brown to a blue-black if there is a presence of the starch in the food sample. The food will change into the color of iodine, the iodine solution will change from brown color to a blue-black. That one confirms that the food contains starch. These are the chemicals the Rwanda Standard Bureau used to say the food contains starch and the food is healthy for the human consumption. If they just count and see there is a certain amount or percentage of a particular starch in the food. You have ever seen in the biscuits, you have ever seen in some of the food, they are written some of the percentages that are present in that food components. And they say that food is healthy for human consumption, all right? So we say that once you use the iodine solution, the color of iodine, which is brown in color, turns into a blue-black, which confirms the presence, which confirms the presence of starch in the food sample. All right? I think it is correct. Now, that was our previous lesson. So we try to underline these two words and we highlight them, the blue-black and the brown color of the iodine solution, which changes from brown to blue-black. It means our food sample contains starch. We also talked about reducing sugars. When I talk about reducing sugars, I mean the sugars which are made up of one molecule or a simple molecule of monosaccharides. Examples of monosaccharides, we have the glucose, we have the fructose, and we also have the galactose. All right? We tested about number 2 was testing. Number 2, that was number 1. Number 2, we have been testing about reducing sugars. We did a practical of it, a reducing sugars. So the reducing sugars, we used the glucose as an example of reducing sugars. How do you know that the biscuits contain glucose? How can you confirm that glucose is present in the biscuits? And you know that what the biscuits contains glucose, and glucose is one of the energy-giving foods. They give us energy for doing work. energy for movement. Particularly in biology, talk about energy for locomotion, able to move from one place to another place. I think you do understand the difference between movement and locomotion. When you talk about movement, it means moving your part of the body, that is changing the position, that is movement. But when it comes to locomotion, that is shifting the whole body from one place to another place. So once you are locomoting, you need energy, you need glucose. That's why glucose is mostly given to the people who are doing more exercise, athletics. They need more glucose. So what do we use to test the glucose? We use the Bendix solution. Bendix reagent is the chemical test of testing the presence of glucose in the food sample. If you want to know that the food contains glucose, you use the Bendix solution. And once you use the Bendix solution, you do what you call color observation. You observe color changing. Color changing. What is the color will you observe? Automatically, the color of Bendix solution is blue in color. And that color, once the food sample contains starch, what will happen? The food sample will change from blue, as I told you, from blue to green, then from green to yellow, then from yellow to orange. And it can also change to brick orange when the amount of glucose is very high. When the amount of glucose present in the food sample is very less, that one it will not change. So quickly, that was a review of the previous lesson, lesson 1. And now we're going to go to lesson 2. The lesson 2 is concerning about testing of non-reducing sugars. As you can see, this is testing of non-reducing sugars as well as proteins. We did a practical lesson. We are going to go through the theoretical part of it. I hope everybody, you have a pen, you have a notebook, try to note what are the procedures, what are the apparatuses required, and what are the reagents we need to carry that experiment. All right? When you have all those requirements, when you know the reagent required, when you know the procedure, when you have the apparatuses, it is very easy for you to conduct the experiment. It is very easy for you to observe the color changing. Quickly, let's go to our lesson of today. The learning objective of today is to state the chemical reagent used to test the presence of non-reducing sugars, and as well as the proteins. We try to highlight or list the suitable apparatuses required for the chemical test of a given food sample. The food sample I'm talking about is non-reducing sugars. Non-reducing sugars. Non-reducing sugars, examples are sucrose. We also have to carry the test to identify the presence of non-reducing sugars as well as the protein in a given food sample. All right? You know the procedures, you have the apparatuses, and you follow the procedures I'm giving you, you will be able to observe and make a conclusion or a deduction that that food contains proteins. That food contains non-reducing sugars. Surely, I'm very happy to share with you that lesson. We did a practical part of it. I'm now doing it a theoretical part of it so that you can note it. Once you get those requirements and reagent, you're able to conduct the experiment and you will get a positive results. And I hope nobody will get a negative results. So we are going to do non-reducing sugars, proteins in the food sample, as well as we give the examples of non-reducing sugars as mentioned, sucrose. Sucrose is an example of non-reducing sugars. Sucrose, if you remember, sucrose is when you get a glucose, this is glucose, all right? The glucose, when you get the glucose and you combine it with, this is a bond, you combine it with the fructose. These two, they will make what you call the sucrose. And they have a bond. Who can remind me the bond that joins two molecules of glucose? That one is called glycosidic glycosidic bond. The bond that joins these two monosaccharides is called glycosidic bond. That bond is the bond that joins to these two molecules. So what do we use to test the presence of sucrose in a food sample? There is no specific reagent that is used to test the presence of non-reducing sugars. We do use Bendix solution, but followed with other chemicals. Let's go to the materials required. We use the Bendix solution, which is brew in a brew reagent. We use hydrochloric acid, which is a colorless. The hydrochloric acid is colorless. All right? We also have what we call sodium hydroxide, which is a base, which is also colorless. We must be having what we call test tubes, and also test tubes. We have test tube breakers. We have source of heat. We need heat. We need droppers. We need beakers. We need water, and as well as we need our food sample, which is a non-reducing sugar. Quickly, let's go the procedures now. How do I conduct that practical lesson? You put 2 milliliters of non-reducing sugars or sucrose in a solution, in a test tube. You add a few drops. We add a few drops of dilute hydrochloric acid, and then you boil. The question is, why do we have to add hydrochloric acid in the sucrose? We have that bond which is called glycosidic bond. That bond is very strong. Once you add hydrochloric acid, it will break the bonds of glycosidic bond, and the glucose and fructose will be separated and form a monosaccharide. You are going to have glucose and even the fructose. Then after you cool the solution. After cooling it, you add sodium hydroxide. Why do I have to? Because once you put Bendix solution in a sucrose only, it tests negatively, because the bonds of the sucrose are very hard. It needs to be hydrized. It needs to be broken down by hydrochloric acid. This bond, which is called the glycosidic bond, after being hydrized, they separate and form a single molecule, which is glucose and fructose. Now it is able to test positive when you add the Bendix solution, followed by cooling and adding sodium hydroxide. Student, I ask a question, why do we have to add sodium hydroxide? The reason why we added sodium hydroxide in a previous practical lesson is to neutralize the acid present in a food sample. Then from there is sodium hydroxide slowly until fizzing stops, and add Bendix solution and heat and shake. Record your observation, and after drawing the observation, you carry out the conclusion. This is the observation you are going to have. Observation, change of the color from blue to green to yellow to orange and then to red. That one indicates the presence of non-reducing sugars. This is the conclusion you can see from here. This is a Bendix test. We are testing it is used for testing reducing sugars as well as non-reducing sugars. You can see this is non-reducing sugars. This is the color is changing from blue to green to yellow and finally orange or red precipitate, depending to the amount of glucose present. These changes in color indicate the presence of non-reducing sugars in the food sample. Let's go testing about proteins, which is a practical 2. You have seen practical 1 was testing non-reducing sugars. Practical 2 is testing about proteins. How can we say that a food contains proteins? All right? Testing of proteins. Proteins are essential nutrients of the human body. They are one of the building blocks of the human tissues and can also serve as a fuel source. If I say fuel source, it means protein can also give us energy. All right? They contain as much energy as carbohydrates, but less than lipids. It means lipid has more energy compared to the carbohydrates and the proteins. So it means that amino acids are the building blocks of proteins. What are the building blocks of proteins? The building blocks of proteins are called amino acids. All right? We have very many amino acids in our body. Almost we have 20 amino acids in our body. And these amino acids, they perform different functions in our body. All right? Most of the food we eat contains these amino acids. Examples, we take milk in our home, we take eggs, we take meat, we take beans, we take peas, are examples of the food which contains proteins. I've tried to show you some of the diagrams that shows you the food which contains these proteins. We have the beans, we have the peas, and examples of the food. These are the examples of the food which contains proteins, all right? I think everybody can observe the two diagrams. This is diagram A, which represents beans. Diagram B presents the peas. So there are two major ways in which the scientists test for the presence of protein in the food sample. We use two reagents that test the presence of protein in the food sample. These are the tests for proteins. We use two proteins. We have two of them. One is called Meron's reagent, Meron's test. The second one is called the Burette test. We are going to discuss about the Meron's test today, and in the next lesson, we're going to discuss about the Burette test. In this test, the reagent is known as Meron's test. It is used to test the presence of proteins in the food. We use Meron's, we use Burettes to test the presence of proteins in the food sample. These are tests that confirms that the food contains proteins, all right? Let's go through the way and the procedures. To test for the presence of proteins by using Meron's, these are the requirements. You must be having test tubes and a test tube breaker. You must be having a source of heat. You must be having droppers. You must be having measuring cylinders. You must be having what you call the syringes, cooking oil. These are also examples where you have cooking oil, we have egg albumin, we have distilled water. I said, why do we use distilled water? Most of the time we use distilled water as a control experiment. Milk and now Meron's reagent. This is the table that shows what will happen. The table below shows the results that may be obtained during the Meron's test. We're going to do it practically, but let's have the theoretical part of it. We have number one, test tube procedures. We have observation, and we have the deduction. You get 2 centimeters tube of the food sample in the test tube, add Meron's reagent, then after adding Meron's reagent, you need heat. You have to heat. After heating, what will be the observation? What will you observe? All right? The observation is here. Once you get what you call a coagulated pink mass appears, automatically that one obviously confirms that my food sample contains proteins. That's what is written there. Once you get that color, I'm emphasizing on that color, coagulated pink mass appears. The proteins are present in the food sample. We did it practically. We are now talking about a theoretical part of it. That is, you note that thing, that if you use the Meron's reagent and you boil, when you are boiling, you observe a color change, which is a coagulated pink mass that appears. That one confirms the food sample contains proteins, all right? When you get 2 centimeters of the food sample in a test tube and add Meron's reagent and boil, the color remains colorless. You get Meron's reagent, you add it there, but in the food sample, there is no change in color. The color remains colorless. What will you observe? What is your conclusion? What is your deduction? My deduction is going to be telling me that there is absence of food, there is absence of proteins in the food sample. Once there is the colorless color is observed in the food sample, that one confirms that that food sample does not contain proteins. All right? Remember, I've told you that there are two tests that we carry out to test the presence of proteins in the diet. One is Meron's reagent, another one is the butyrate test. All right? Let's go to before we go to assignment. Before we go to assignment, let us remind ourselves what we've discussed today. We have discussed about non-reducing sugars. We have talked about how reducing sugars is tested. How do you observe the food contains non-reducing sugars? I said there is no specific test for non-reducing sugars. We use the Bendick solution, and once you use the Bendick solution, which is a test for reducing sugars, followed by hydrochloric acid. Why do you add hydrochloric acid? To hydride the bond. And after hydrazing the bond, that bond which joins the two molecules of sugars will be broken down and form a simple sugar, that glycosidic bond. And now you add sodium hydroxide to neutralize the acid, and now you heat. When you are heating, you have to observe color changing. We have talked about proteins. I said the protein can just be tested by using two tests. One is Meron's reagent, another one is the butyrate test. And you have seen what you observe. So quickly, let's have the lesson assessment of today. This is, I am going to leave with you 3 numbers. Number 1, the DASH tests uses several reagents. The reagents are mixed in equal proportion, DASH, and the DASH makes up this reagent. Number 2, the bonds that holds protein structure are known as DASH. The 2 tests for proteins, 2 reagents are used, Meron's reagent and the butyrate test. Which of these two tests does not involve heating? So student, I'm asking which of these tests which does not require heating? So student, thank you very much for your attention and participation. I wish you a good day and stay safe. May God bless you.